I'd now like to welcome the last speaker of the program, Dr. Craig Venter, who is the founder of the J. Craig Venter Institute. Close to a quarter century ago, Dr. Venter led the team to decipher the sequence of the human genome. Although this incredible feat opened the door to a host of knowledge, there is still much to be learned on the role of our genetic code in shaping our phenotypes, disease risk, behavior, and even our personalities. Dr. Venter maintains that, especially in Western practices, clinical medicine is based on resolving symptoms which are often too late or very expensive for long-term survival. To circumvent these outcomes, he has developed programs to determine comprehensive phenotypes, together with detailed individualized genome analysis and advanced screening techniques. We will now hear more on these advancements, which have shown great promise in non-invasive means of improving early detection of health-related issues. Welcome, Dr. Venter.
Well, thank you, Lori and Bent, for having me here. And I think you'll see why they chose me to be the last speaker, because I'm the least likely person here to become a scientist. When Professor Yagi was admiring crystal structures at age 10, I was increasing security at San Francisco Airport by racing airplanes on my bicycle. And the police would come and I could get away through a ditch after a couple weeks, and I could beat the airplanes for the first 100 yards or so. I came back and they were building a fence. So I was the start of airport security. Because I have a condition called aphantasia, where unlike most of you that probably have photographic memories, I don't see any pictures at all. So I think conceptually, not in terms of pictures. So for lots of reasons, I barely graduated from high school. At age 17, I moved to Southern California to take up a surfing career. But this was in 1964. Most of you don't remember that time, but we had a war going on in Vietnam, and I got literally drafted off my surfboard in 1965. I ended up in the Navy Medical Corps and ended up as a medic in Vietnam, where I was like a sponge absorbing medicine treatments. In the military, you can do whatever you're trained to do, and they trust you to do. So as a 19-year-old, I was teaching interns and residents how to do spinal taps and liver biopsies. I turned 21 in Vietnam. I was doing major surgery, and I was a doctor for a village and an orphanage for a year. And it was very satisfying work. I'm one of the few people probably that came out of Vietnam better off than he went in. I wanted to go into medicine to practice third world medicine, but I had to start school over from scratch. So I started at a community college in San Francisco and then transferred to UC San Diego. And something happened once I got there. I was introduced to high-end scientists for the first time in my career. And I think because of my unusual background, some of these scientists took a liking to me. I was given my own research lab as a junior with the late famous biochemist Nate Kaplan, who trained with Fritz Lipman, and I spent time with Lipman as well, just for the lineage Lippmann trained in Germany with Meyerhoff, and you've learned about the Meyerhoff pathways. So I have a very good lineage. Lippmann wrote a book called The Wanderings of a Biochemist that had a lot of influence on me of the strange route he followed through science. I made what was considered a major finding at the time and published my first paper as an undergraduate, and I proved that adrenaline worked on the outside of cells, not inside. And all of a sudden, I just fell in love with doing science, changed my plans from going into medicine, went into science. I hold the record for the shortest PhD for the University of California system. Didn't do a postdoc and went on to do research, ending up at the NIH intramural program, where I started exploring life from a different point of view through the lens of what became genomics. I had the first automated DNA sequencer developed by Lee Hood's lab and company. And I sequenced one gene, and I thought this was the most tedious, silly approach to the world. So instead of searching for the 1 out of a million clones, I developed a simple procedure of just randomly selecting clones off a plate and sequencing them. And it became very upsetting to the world because we were discovering thousands of new human genes every day. Professors at Harvard and a place we call the NIH director trying to shut down my lab because it was unfair that I was making these discoveries so quickly. And I was messing up their PhD students' theses by finding the proteins that they were going after. The latest count, there's up to 74.2 million ESTs now in the public databases. This was the entire means that the human genome was annotated with genes and still is today with all the splice forms. We had to develop new algorithms to deal with this large number of sequences. And my friend and colleague, Ham Smith, who was pictured in one of the photos the day of his retirement, decided we could apply this mathematical algorithm to sequencing a genome. So we sequenced his pet genome, Haemophilus influenzae, which is what he isolated the first restriction enzymes from that led to his Nobel Prize in 1978. This ended up being the first genome sequence in history. That wasn't our goal. Our goal was just to test the idea, and it ended up working really well. But the problem was trying to define life at the genome level is that over 40% of the genes that we discovered were of unknown function. And so one of the notions we had was by sequencing lots of other genomes, we could maybe find these by comparative genomics. So over the next several years, we sequenced a large number of species, dozens and dozens of pathogenic bacteria. The first archaea went on to do Drosophila and eventually human. And then we did 65,000 miles sailing around the ocean, filtering seawater and shotgun sequencing everything in the ocean, and we discovered literally tens of millions of new species. Erling Norby wrote the foreword to this book. This is the US version. The British version is called Microlands, but it's the same story, and you can get that. But again, most of these genes didn't look like anything anybody had seen before. We found huge families that were highly conserved, but it was very unsatisfying. So we decided maybe the only way to understand the genome was to take the Feynman approach and build it ourselves. This project took over 10 years trying to design life for the first time. And we had a contest at the Institute for people to put their designs together. We made 3 different designs. Not one of them led to life. And it's because we do not know enough biology to design things on first principles. It took us a very long time adding genes back randomly until we could get life and then minimize that. And you can see in the numbers here, out of the smallest genome of any species, this created a whole new life form that didn't exist. But 149 of those genes, essential for life, are of unknown function. Nothing is known about the biology. Now, when we look at the human genome, it's even a much larger number, larger percentage. So despite discovering tens of millions of new species, having more sequences than probably anybody ever thought was even possible, it wasn't increasing our knowledge substantially just looking at sequence. When the first discussions about the Human Genome Project took place in the mid-'80s, I got immediately excited about it because I had spent 10 years trying to get one protein. So the notion of getting them all in 15 years was very exciting. And then we realized the approach that we developed for the first genome could speed this up. A company called Applied Biosystems called me up and offered me $300 million to sequence the genome with their new machine. I thought they were joking, but eventually we settled on it. And we sequenced the genome in 9 months using the shotgun sequencing approach. And the challenge has been to then understand how this codes for each of us. That was a haploid genome. A few years later, Sam Levy led the team at the Institute to do a complete diploid genome. It was actually my genome, and it became a well-studied. But the key thing about the diploid genome is 75% of the variation in the genome is not contained in SNPs. Everything that's done in genetics today is looking at the 25% of single nucleotide polymorphisms because we have not been able to do diploid genomes. To get diploid genomes here, we had to sequence multiple haploid sperm cells to get phasing of the genome. Now with PacBio and nanopore long-range sequencing, we can assemble complete diploid genomes. Having lots of genomes, we could ask unique questions about the structure, and some of the interesting things we found. Every place you see these dips down, this is looking over 1 billion data points in the genome, are places where the genome cannot tolerate any mutations. So those places where it goes down to 0, if you have a mutation there, those would be things that would cause a spontaneous abortion, for example. They're just incompatible with life. But you see lots of variation. We pooled all the known transmembrane domains together, and you can see there's quite a dramatic difference along these transmembrane molecules. The portion that sits in the lipid membrane can't be messed with. If you get a mutation that puts a charged amino acid in there, it would be a loss of function mutation because the protein would have been sped out of the membrane. Other places you can see it doesn't matter very much. Now, the other finding that we made early on was that mice are not little humans, and human cells and culture are not good representation of humans. So this is looking at essential genes that could be characterized, and that when you looked at the essential genes set in humans, that's where you see primary pathogenic disease. So there's lots of genes that are non-essential, and then we have the whole set that we have no idea what they do, but we start to see pathogenic genes. Now, my genome had gotten well characterized by a lot of people, and there are a lot of reports on how I was a heterozygote for ApoE4, and then, according to the literature, had a greatly increased chance of having Alzheimer's disease. So I asked my friends in neurology, in radiology, would they do a PET amyloid MRI and a MRI brain scan? And they said, we can't do that because you don't have any symptoms. And this is a theme we'll keep coming back to. They finally agreed to do it under a pseudonym because they were worried the genetics would accurately predict things, and if they found that I had early developing Alzheimer's, that it would ruin my career. So they came up with a pseudonym. I suggested Charles Darwin. They said, that's too obvious. We don't like that. And so they agreed and did a brain scan, and we were on vacation the following week, and they called to report the results. They were very surprised. They said, you have a brain of a 40-year-old, and the amyloid was totally zero. My wife, Heather, said, no, you're wrong. He has a brain of an 18-year-old. But that's a different situation. But it became clear there was a dissociation of what the genetics community was reporting and what we found by actually doing a real phenotype assay. So it became very clear to me instantly that we weren't going to understand the human genome by just sequencing thousands and thousands of human genomes, that we had to have comprehensive phenotype information on each individual together with their complete diploid genome. And we had to have this thousands and thousands of times. So we set up a system with the goal of doing non-invasive phenotype other than for a needle for a blood draw, something that could be done in less than 8 hours, and set up a paradigm to do this with only starting to look at people that were self-proclaimed healthy. And so you can see we're just trying to understand the genome, trying to set up genotype-phenotype correlations, and the world started to change very dramatically with just trying to do the simple basic science approach. One of the key things is we were using a new tool in MRI imaging called restriction spectrum imaging. And this is a game changer, even though it's been around for 15 years. It hasn't reached standard practice yet because medicine is very slow to change. With restriction spectrum imaging, it looks at water molecules. And it turns out solid tumor cells like prostate cancer cells have larger nuclei. They have more water molecules, but the water is less mobile. So the tumors, because of the signal, light up like light bulbs. Now, I don't know if you can see this, but the bright orange areas are where the restriction spectrum imaging indicates that there's a tumor in the prostate, and the ultimate pathology agreed totally with that. The very first individual to come in to this phenotyping clinic was a physician and his wife. They were going to go on a long vacation the next day, and he just thought it would be good to have a physical. And you can see this bright light. That was a tumor found right under his breastbone. We ruined his vacation plans, but he credits us with saving his life. So starting with individual one of a healthy individual, it wasn't so healthy. I was testing the MRI machine after being told by one of the top urologists in the world after biopsies and MRI testing that I did not have prostate cancer. But my own radiologist, after testing the machine, came and told me that I had 2 very high-grade prostate tumors, and that they were biopsied and confirmed, and I had surgery right away. And it's known, we spent a lot of time trying to analyze my genome, doing the genotype-phenotype analysis of why I would have such high-grade prostate cancers. So the adrenaline, the androgen receptor, testosterone receptor, is linked to prostate cancer because the prostate cannot grow without testosterone. And so there's lots of things that have that associated with cancer, and one of the major treatments is basically removing testosterone from the body. The prostate can't grow and cancers generally stop. Looking at the androgen receptor, it's a triplet repeat disease gene. It's on the X chromosome, so men get 1 copy and women get 2. And there's a series of CAG repeats in exon 1. And it turns out there's a range of 7 to 36, that's sort of a normal range. The higher number of repeats, the less expression of the androgen receptor there is. Individuals with very high numbers of repeats, 40 to 62, have, there's 2 different diseases. Klinefelter syndrome shows up, and Kennedy's disease with this big expansion. There's also an unusual phenomenon. The higher number of repeats you have, the higher the odds of you being left-handed. Lots of things can be attributed to the X chromosome and to the testosterone receptor. The opposite is true with the lower number of repeats. The fewer repeats you have, the higher the expression of the androgen receptor. On sequencing, looking at the sequence of my genome, I have the lowest number of repeats thus far demonstrated. I had only 3 CAG repeats. So I massively overexpress the androgen receptor. On having endocrine analysis, it turned out I had very low testosterone levels. But when you have massive amounts of receptors, it just takes a small amount of testosterone to stimulate that. So even with low testosterone, I had a high testosterone phenotype. I was bald by age 21, risk taker, aggressive personality. But because I had low testosterone levels, the endocrinologist put me on exogenous testosterone, which was the worst thing to do. But they weren't looking at receptor levels of the genome. So now I had tons of excess testosterone with tons of excess receptors. And that's how my tumor developed very rapidly. I actually had 2 tumors. We grew the tumors in culture. They literally grew 10 times faster in the presence of testosterone. So that's a story of my own, of genotype-phenotype analysis, and it's indicative of what we could do with individuals of tracing back to the genome in many cases to find the genetic association with the disease. I'll show you several other pictures just to show the power of the restriction spectrum imaging. I paid to have all the scientists in my institute go through. This is an image from Ham Smith when he was 84. He had no symptoms. We found a massive lymphoma growing in his chest that if we had not found it, the surgeon said he would have been dead in 6 weeks. He turns 93 in August, and so he's doing well. He has a cough from radiation damage from the treatment. But you can see a 10-year-old could do this diagnosis because of the RSI imaging. Here's just another example, a similar type of tumor. Here's a young woman with a thyroid nodule. Again, the tumors just light up very clearly. It was confirmed by biopsy. With brain scans, quite often we see brain tumors. This one may be a little bit harder to see. It's that strawberry type thing at the top of the skull. Keep in mind, every one of these individuals are asymptomatic. They thought they were perfectly healthy. What we find in healthy people is a high-grade tumor in everybody over, in 5% of everybody over 50. So fortunately, there's a lot of young people in the audience, but I'd say there's several here that are over 50. 5% of you probably have a tumor that you are unaware of. The MRI also gives us amazing metabolic information. It's one of the few ways to measure organ fat, and it comes right off the MRI image. So we diagnose a large number of people with metabolic disease. It turns out with organ fat, I was on a 30-mile bike ride with the world's expert on organ fat, and we stopped for a break halfway through, and he said, you know, doing all this exercise won't change your organ fat. Only calorie restriction will. And I said, well, why didn't you tell me that 15 miles ago? But it's very key because people that develop organ fat go on to develop NASH, quite often need a liver transplant. We can use the data from the MRI to get very quantitative information of all your muscle mass and your peripheral and visceral fat as a measurement. And it was really quite amazing in this group of asymptomatic, healthy people. The fatty liver detection rate was 23% across all age groups. We also do non-invasive cardiac testing with echocardiograms, coronary CTs, and remote monitoring. This is what the images look like. You can see they're incredibly accurate, where you can get very precise measurements of the heart, diagnose things. The valves show up very cleanly. This is all with non-invasive echocardiogram. With CT scan, we see calcification plaques at around 12% of people, half of them under 60. And again, these are people that are asymptomatic. Getting back to Alzheimer's, like I thought that I was at risk for, we've screened now a large number of ApoE heterozygotes. Not one have we found Alzheimer's disease in. In contrast, we see correlations of about 80% of homozygotes with Alzheimer's disease, but it's not 100%. This is the type of volumetric assays we get. If you can see the yellow areas, though, that's the hippocampus. And so the main indication, you can see the loss of the hippocampus on the right. So it's fairly easy looking at the volumetric from these non-contrast brain images. But here's a 73-year-old homozygote for ApoE4 with a completely normal brain, no hint of Alzheimer's. So the genetic studies that are done are generally looking at very specific populations with disease, and then they over-extrapolate to, well, if the homozygote has the disease, heterozygotes must be at risk. The same information, too much extrapolation was done with BRCA1 and BRCA2. If you have genetic changes in BRCA1 and BRCA2, you only have a 50% lifetime risk of breast or ovarian cancer unless you have a tremendous family history. If every woman in your family has breast or ovarian cancer, the risk goes up in the high 90%, which is what physicians were telling people just based on the BRCA1 and BRCA2 data, which was totally false. But it means that BRCA1 and BRCA2 are not the cause of breast and ovarian cancer. They're linked to it. And the family history shows that the genes that are actually causal are yet to be discovered. Again, with no contrast, the MRI images give us a complete view of your vascular tree. You used to have contrast to get these kind of images from the brain. And what we started seeing again in the healthy people, I don't know if you can see that little bleb there at that loop. That's an aneurysm. We find aneurysms in 1% of the population, but mostly in people under 50, which means genetic selection has already taken place. You rarely find aneurysms in people over 50 because they've already died from them. Aneurysms can be treated in 15 minutes as an outpatient, but generally what happens that people show up in the emergency room with a massive brain bleed and die. So these individuals, again, are all asymptomatic. These are just straightforward assays with people with no apparent disease. They exercise, they're certain that they're the healthiest people on the planet. Physicians don't like doing these assays, and the reason for that is in the 1970s, when CT scanning really became popular, CT is just X-ray. Clinics were set up, even storefront clinics. There were lots of whole body CT scans done. CT scans cannot distinguish between a tumor and a cyst. And so it meant that further studies had to be done, usually a biopsy to see if it was a tumor or a cyst. Insurance companies complained that it was increasing their costs. Patients were put through all kinds of unnecessary procedures. So since the 70s, medical students were taught that it's basically unethical to do tests on asymptomatic patients. Fortunately, the technology has changed. And now with this new type of non-contrast MRI imaging with restriction spectrum imaging, and you'll see adding AI on top of it totally changes it so we don't have so-called incidentalomas anymore. The challenge is teaching the existing medical profession about these changes. We had an extremely wealthy individual that we diagnosed as having a renal carcinoma. He went to Stanford University to get a second opinion. They did a standard MRI, told him he just had renal cysts and not to worry about it. We convinced him to see another radiologist at UC San Francisco who we knew understand the RSI imaging. He confirmed our diagnosis and insisted he get a biopsy. Turns out he did have advanced renal carcinoma, and if he'd listened to standard medical care at Stanford, he probably would not be alive today. So we stumbled on this. We were doing basic science that led us in totally a different direction. The United States has probably the highest healthcare cost in the world. Almost, I think, 5 to 10 times what they are in Sweden and Norway and most other highly civilized countries. But in the US, and I think these numbers hold up pretty much across the board, 1/3 of you that live to 50 will not live to age 75. And the key reasons are death from cardiovascular disease, cancer, and then accidents is the third leading cause of death. More than 75% of all the healthcare costs in U.S. are due to these few diseases: heart disease, cancer, stroke, metabolic disease. And these are also, as we've shown, are the most preventable with early detection. So the effort is to try to get the costliest 10% of the population, which accounts for 63% of the cost, early diagnosed. 1% of all the patients count for 20% of all the cost. And I'm sure that's true regardless of the country. You just do it slightly more efficiently and the care costs less. People with 3 or more chronic diseases are in that 1% category and cost the most. Diabetes is the single biggest cost as a chronic disorder. But these things can be changed. I don't know if this is big enough that you can read it. This is the latest statistics on deaths per year in the United States and the difference between the health span and the lifespan. The ones in red are all the diseases that we detect with our non-invasive 6 to 8 hour testing. And with early diagnosis and early treatment, we convert these chronic diseases into acute treatable disease, and the cost goes down very substantially. And we think we will narrow the gap between the health span and the lifespan. This is longevity of all males, all races in the United States. And you can see there's a huge disparity. The South is much less healthy. And then you see patches of red around the country. Those are primarily Indian reservations, where, again, there's much shorter longevity. Genetics helps. If you have 2 X chromosomes, the world looks much better. But even in the South and in the Indian reservations, they still stand out as shorter longevity than the rest of the country. But there's tremendous socioeconomic and ethnogeographic disparities. For example, American Indians, for males, the life expectancy is only 62 years. For the average for Black Americans is 67 years, but in cities like Washington, D.C., it's 55 years. So there's different pockets and different places. So we have proposals pending to create mobile clinics, mobile MRI and mobile cardiac clinics, going to indigenous populations and to some of these areas with tremendous health disparities based on race and socioeconomic factors. Now, these tests are pretty inexpensive to do. The biggest single cost is the cost of radiologists to read the images. Because to do whole body MRI plus the brain scan, it takes 4 different radiologist specialists. Now, radiologists are getting scarce because they're afraid AI is going to replace them, which we're trying to hasten that. So because there's fewer of them, the cost keeps going up for radiology reads. But we think that can be the single biggest impact in the cost of this preventative health care is getting AI to read the MRIs. Now, with solid tumors, the restriction imaging, all of you can diagnose those just looking at it. You see the bright lights. With soft tissues, breast and intestine, it's much more difficult. We're working with one of the world's experts on breast MRI imaging. We're trying to apply AI to this, and the Venter Institute is going to open a new clinic dealing with this, with using imaging primarily for improving women's health. But AI is absolutely essential to get away from mammograms. Mammograms, as you know, are just X-rays, so they have all the problems that the CT scan had. They can't distinguish between cysts and tumors. With this new technology, we think we can get there. So it's like Lippmann's route. We started out in one area, started out to do an experiment to improve the interpretation of the human genome. And as I said, in individual cases, we're doing this collectively and using AI to add the phenotype to the genome. We think we're going to make a lot of progress in a short while on how each of our genomes codes for us in our conditions. But I think this has a chance to be an exciting change for the future. My calculations are it could save the US government $2 trillion a year in healthcare expenses. But turning how medicine is practiced and paid for upside down is probably the biggest challenge we could ever face. But thank you very much.